episode 205 of the Stem Cell Podcast, Stem Cells in Space, with Dr. Kate Rubins. Hey, everybody, we are Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds on Earth and beyond. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we have Dr. Kate Rubins, a NASA astronaut and microbiologist, on the podcast to talk about her work carrying out stem cell projects in space, some of which were designed by my co-host, the intrepid Arun Sharma. We've also got our usual roundup of recent highlights in stem cell news coming right up.
But first, and definitely can't wait to catch up with Kate, we'd like to introduce Muscle Cell News, a free weekly newsletter provided by the Stem Cell Science News Program. Muscle Cell News summarizes all of the latest research, news, jobs, and events in muscle cell research and delivers it right to your inbox every Monday. Save time and keep current with Muscle Cell News, and you can subscribe for free at www.musclecellnews.com.
Arun, I'm starting off the roundup talking about something. I'm like bored with myself, but I can't not talk about the blood. This is a big one, so you're gonna have to forgive me. We've been searching for the holy grail of, I'd say, medicine, but specifically hematology and hematopoietic biology, the hematopoietic stem cell, right? That's what we've been looking for decades, since we knew it existed. And we think we're there. Every year you hear a story. Oh, we got it. We got it from iPS cells. Oh, we can self-renew it from the blood. Boom, boom, boom. No, it's falling short every time. We're not quite there. The thing is that at steady state, the hematopoietic stem cells are quiescent. I mean, that's the whole thing. They got to hang around for the entire lifespan, right? At least a store of them as they're mobilized under stress conditions and to maintain physiological balance of the hematopoietic system. But in order to maintain that quiescence, they're they're on the low, exhibit very low biosynthetic activity. And in order to do this, they rely on, it's thought and largely agreed upon, they rely on glycolytic ATP production, thereby inhibiting mitochondrial oxidative phosphorylation that results from mitochondrial activity, so that they can hang around for decades, right? But they also have to be able to reversibly switch the program under stress and meet the high energy demands of when you mobilize a hematopoietic system and you make billions of cells from one, right? So this has been the question. How do you mobilize? Not so much how do you mobilize, but how do you retain a hematopoietic cell in the quiescent state? And it's emerged over the last few years that metabolites and metabolism is key here. Metabolites can be detected by transcription factors, and then those transcription factors can modulate all kinds of gene activity. And vitamins, as metabolites in particular, have been suggested to play important roles in stem cell function. And in previous work, actually, Nina Cabezas-Walshid, who's now at the Max Planck Institute, in previous work, she showed that the vitamin A retinoic acid played a really critical role in hematopoietic stem cell function. In fact, that if you deprive vitamin A from mice, they lose quiescence. And if you treat them with this vitamin A metabolite, this active metabolite, all-trans RA, all-trans retinoic acid, you can preserve stemness, even under stress conditions. But the way that this system specifically and generally, the way that retinoids regulate HSC activity is really largely unknown. And so that's where Nina came in here and her group, and really leveraged the real technical developments. We're able to do such amazing things now with all the omics. The scientific community at large is able to do amazing things with all the NGS and metabolomics, etc. And Nina's group leveraged those resources here to get over this major technical challenge, which has really been central to HSC biology is their scarcity. It's so hard to get them, right? It's so hard to get them in abundance because they're so scarce. So that's really been a limitation in trying to understand what the interplay between all these different, biological apparatus are: metabolism, the transcriptional activity, the epigenome. So here they went omics to the max with low input metabolomics. They did RNA-seq, they did ATAC-seq, they did ChIP-seq. And with that, those four horsemen, they revealed these critical metabolic hubs that are specific to hematopoietic stem cells. And indeed here, using those tools, and this is what's great, I think, about leveraging all these next generation tools, is that it's discovery, all the discovery that they give you access to. It's not just about proving your hypothesis, but stumbling upon stuff and seeing how central it is. Here, they uncovered that there's this non-classical retinoic acid signaling axis that relies on CYP26B1. And while in the traditional view has thought that CYP26B1 was limiting of the effect of retinoic acid and retinoic acid signaling, they show here that CYP26B1, in contrast, is indispensable for making this active metabolite 4-oxo-RA. And mechanistically, they show that 4-oxo-RA acts via this retinoic acid receptor beta. But here's the key. They show that this single metabolite, or they emphasize, as they say, that this single metabolite controls stem cell fate. Okay, and that's a landmark and a bombshell. And I'm just waiting for all the hematopoietic big gurus to get in line to chop this paper down. But I mean, from where I'm standing, this looks like a real critical insight into understanding how hematopoietic stem cell function is governed, mobilization is governed, quiescence maybe, and maybe a means of generating hematopoietic stem cells and keeping them in this or multipotent state so we can maximize their therapeutic efficacy. So I'm really excited about this, but I'm just waiting for all the stones to take flight.
Yeah, another reason to take your vitamins, to eat your carrots, to help maintain your HSCs, right? And certainly a beautiful story with combining a bunch of omics analyses, metabolomic analyses, transcriptome, epigenome, to really get to the heart of why this pathway may be able to influence HSC renewal. Yeah, I mean, I think that's the key point, right? Anytime you're able to distill these pretty complex processes down to a single metabolite, a single entity, and like this CYP26B1, you wonder if it's really that exclusive. And I'm of the opinion, and probably, my gut feeling is that it's not that simple, And so I agree with you. I think it's just probably a matter of time before folks start to realize that it's probably a little bit more complicated than just the single metabolite, right?
Yes, absolutely. But, again, to emphasize the approach, I think that this representative of a new era. And I think all the papers we're talking about today, as we always do, because we're at the cutting edge, Arun. But this one and the others we're talking about today in particular really show how the tech is informing discovery. These are no longer experiments that are born of the mind of some seasoned researcher who's had hypotheses kicking around for decades. The tech can lead you to the light, and I think that's a really big deal for younger researchers looking at science and just seeing there's so many stones that are sitting right in plain sight. We just don't really have the resolution to see them.
Yes, absolutely. For the young scientists out there, there's no shame in latching on to a cool new piece of tech and making it your own, because there's so many different pieces of technology that you can really learn and take to heart, and it'll certainly help your career in that way. One of those is all these omics technologies, right? So the epigenomics, metabolomics, you can kind of turn that into your own skill set. And certainly another one that we love to talk about here on the show are these brain and cortical organoid cultures, as we've discussed ad nauseum through many papers and many guests in the past. And yeah, so I'm going to shift a little bit from the blood to the brain and focus on one of these papers coming out in Nature Neuroscience. It's actually utilizing brain organoid slice cultures, kind of a different twist on the organoid story, to look at some of the mechanisms driving ALS, right? That's amyotrophic lateral sclerosis. It's a fatal and currently untreatable disease that's characterized by really sad, debilitating, rapid cognitive decline and ultimately paralysis. This is, side note, this is actually part of the work that my department chair here at Cedars-Sinai, Dr. Clive Svensson, is focusing on. And yeah, we need to really get to the bottom of this disease. It's so sad to see the progression of this particular disorder. So we got to figure out how the cells that are contributing to ALS are, making this disease progress, and that's critical to actually ultimately understanding therapeutic development and development of different targets, different drugs that may be able to reverse some of these phenotypes that are associated with patients with ALS. But the tricky part is, as you might understand, getting samples from folks who are pre-symptomatic, who may be inclined to develop ALS from the brain, that's not really feasible. It's kind of the same story with the heart. That's part of the reason that iPS cardiomyocytes took off, is because it's really hard to get primary cardiac samples for basic science studies, just like it is with the brain, right? And so here what they did is they created a cerebral organoid slice model that's actually created from human iPSCs that can recapitulate some of that cortical architecture that's found in patients with ALS. And it can even display some of the early molecular pathology of ALS that's driven by C9 or 72, which is one of the major drivers of this disease. And kind of like what they did in the last paper, they used a combination of single cell RNA sequencing, different omics approaches, different biological assays to figure out that there are a variety of transcriptional, proteomic, and DNA repair disturbances that are actually found in multiple cell types in these slice cultures, both the astroglia and the neurons too. The astroglia actually have increased levels of autophagy signaling protein, P62, and that the neurons have an accumulation of dipeptide repeat poly-GA and a bunch of DNA damage, different phenotypes that may be driving some of those ALS phenotypes, right? And I guess part of the reason that this is a Nature Neuroscience is that they're able to rescue some of these phenotypes in their in vitro cultures through a pharmacological rescue by a small molecule GSK 260-6414. They got to think of a better name for that one. So yeah, it's another in vitro disease model utilizing cortical organoids, brain organoids in a different context. This is a slice culture, so perhaps you can reproduce and maintain some of those intrinsic cellular connections to get a better understanding of what's actually happening in this setting of ALS. And really, as I said at the start, you need to figure out, Daylon, I need to figure out, everybody needs to figure out what's going on in ALS, because it's such a devastating disease. And just to see the progression of these otherwise healthy patients down this road is, it's really sad to see. So everything helps, right?
Yeah, one of the great promises since early days has been treatments for ALS, because, it's so stark, the difference in these people's lives 5 years on from diagnosis. It's tragic. This one talks about recapitulating mature cortical architecture, but you always, bemoan the struggles we have with getting mature cells to recapitulate phenotypes that have a late onset here. My takeaway from this was that while that is still perhaps an issue, that maybe there is some element of the ALS or frontotemporal dementia, frontotemporal, whatever that one is. Frontotemporal, yeah, that's it.
FTD. You could say
FTD.
There you go. There you go.
There it is, that one. That they may be some pathological hallmarks that are evident even in early stages or prenatally. Am I way off there?
No, I think that's the right idea. It's because this is being driven by some sort of genetic mutation. The thought is maybe you can anticipate the development of that disease phenotype using these cells that maybe are able to be matured more rapidly in an in vitro setting. It is definitely a valid concern, and it's a concern across the board if you're looking at adult diseases. Like, I run into this in the heart as well. If you're trying to study an adult cardiomyopathy, how do you model it in an immature cell type like this, right? I mean, the thought is, if you can distill some of those mechanisms down to a single gene, then maybe you're able to better elucidate some of those processes. But it's tough. It is definitely a tough game.
Well, I mean, with the diagnosis of ALS being early in many cases, this would go a long way if you could address or mitigate or delay the phenotype. So exciting work and important insight. I'm staying in the brain. I'm going earlier even, and this is really earliest, I would say, and important because it's also talking about fundamentally about organogenesis. And this is getting back to our brief little foray there, discussing how tech is really propping up a whole new era of science. This is a great example of that too, I think. When we talk about development of any organ, I think stem cell biologists, or at least the dinosaurs like myself and those older than me, not you, but you're always thinking about cell induction and morphogen gradients and it's a signal that dictates fate. But what we have always appreciated, but I think maybe forgotten, it's taken the younger generation to bring us back, is that it's a mechanical process, right? And particularly in the brain, development begins with folding, right? Folding into a tube. And fundamentally, a failure there in neural folding is one of the most common birth defects out there. 1 in 1,000 pregnancies, everybody knows about failures in neural tube folding because we see it. And there's been animal models to try and address it on the cellular level, even. But there are really human specific aspects of neural tube development that can't be recapitulated in these models. Of course, I hear you're about to say it, organoids, right? And organoids are key. And they've made a lot of critical steps in our understanding of cell fate decisions and the 3D mechanical process underlying that. But they're kind of, I wouldn't say random. I would say it's difficult to reproduce the architecture, anatomic architecture. And the architecture isn't always, I would say even rarely, while the rudiments are there, it rarely approaches, I would say, the superstructure of the general, cell fate patterning that you see in vivo. Then you have the organ on a chip, right? So you can try and enforce some physical restraints. But the problem is the cells are forced into that assembly, which is static. So the cells they lose a lot of the elements of self-organization, right? So this being the case, that background, the group of Sebastian Streichman, who's at in the Department of Physics, all right, and also in the Department of Science and Engineering, Biomolecular Science and Engineering at UC Santa Barbara, and that says a lot. This is a physics paper, although it's in Nature. Inspired by the physical process of in vivo organogenesis, what Sebastian's group did is they did a kind of hybrid. They started with a two-dimensional sheet modeling the neuroepithelium and the lumen below it that's the nucleus of neural tube formation. And so they had a structure that modeled that two-dimensional sheet, and they use that as this kind of biochemical, molecular, and physical starting point. And then they pretty much dumped some matrigel on top of it. And that triggered this 3D morphogenic process, but starting from this initial shape. And doing that, they were able to recreate neural tube folding using human stem cells. And this is a key here, folding with 90% fidelity. I think a lot of these papers, we talk about making what do they call them again? The eye blastocysts,
gastroloids, all those things.
Gastroloids, everything they're making. This is key. And some of them do better than others, but I think it's really important now that we're incorporating the percentages here, because a fluke can happen now all the time. Showing this happens with 90% fidelity, I think, emphasizes that we're approaching a level of physical control here. That's critical. And the end product anatomically resembles a human neural tube, guys. And showing what it took here, making it a Nature paper, they show that it's the neural and non-neural ectoderm are necessary and sufficient. So just those two. And it results from two mechanisms, essentially, is the apical contraction of the ectoderm, and then there's the adhesion of that ectoderm, the basal, at the basal level, to matrix that's synthesized by the non-neural ectoderm. So one cell's creating a matrix, the other is attaching to it and having this apical constriction or contraction. Then they use drugs to show that they can get molecular defects or morphological defects that are similar to the neural tube defects that you see in vivo. I don't know about whether those drugs are really relevant, although I just don't know. They probably are. And lastly, and I think this is key, they showed that it was the width, and this is kind of unexpected. It was the width of the tissue that dictated the shape. All right. So I love this story because it recapitulates a natural process. It derives some insight about what the mechanism is there, and then it provides a little unexpected twist here, showing that it's the geometry, not just the morphogen gradients, that really dictate some of the superstructure of these organs during organogenesis.
Yeah, I really like this story for a few different reasons. One, like you said, the percentage, the fidelity of this process, 90%, that indicates that this is a reproducible thing, right? And if it's really that reproducible, then perhaps other labs can pick up on it and use this as a model system for neural tube development too. And I'm a big fan of that. Two, this is sort of in contrast to the previous story we talked about, right? When we were looking at adult phenotypes for ALS. In my opinion, this actually may be the best way to utilize induced pluripotent stem cell derived cells, is to look at early developmental processes, because when you're differentiating these cells in a dish, you are recapitulating some of those early morphogenic processes, as opposed to an adult phenotype. I'm probably going to set off a bunch of red flags around the community by saying something like that, but I guess I sort of am an adult cardiac biologist, but I still think that the most powerful application for iPSC derived cells is in the early developmental modeling, the early developmental processes. And you're right. I mean, the fidelity is great. The dev bio is great. And the next question for me is, can you use this as in a high throughput way? Like, some of these chip-based systems are not always amenable to like a high throughput format, but they actually kind of alluded to it here. They started introducing some drugs that may be able to recapitulate some of these neural tube defects. So, if you can create some sort of high throughput system for analyzing neural tube defects, I think that would be very exciting.
Yeah, well, this is high profile because neural tube defects are such a high prevalence, right? And at the same time, there's not a lot of defects that present at birth that manifest in like the real primitive organogenesis. So you could even argue here primary. So um, yeah, this is, I think, a unique case, but I'm not disagreeing with you. I agree that early developmental processes are it's where we can really have the strongest insight, because we can control those systems, because they're relatively I don't want to say simple, they're less complex. But for me, the other insight here, and I think it's coming out in a lot of papers that I've seen, is that it's really about cobbling together the rudiments and then letting biology take over. I mean, there was Matthias Lutoff, which was also relatively not a super complex system, but I can envision in the future where you put 10 cell types together, maybe. And if you are able to, even you don't maybe even have to put them together in the perfect micro pattern lattice or anything. But the point being is that combinatorially, I think as we approach a greater understanding, we can integrate more complexity and maybe get to some more end stage organs, some of the organs that take place, post gastrulation, that are a little bit more complex and therapeutically relevant.
Yeah, absolutely. It's a good point to bring up Matthias Lutoff, because he's also very excited about using some of these scaffolding technologies to study developmental processes. So don't forget to think about some of these really cool technologies and how they can intersect with early development and help us understand early development. And so, speaking of actually early development and also neural the neural system, the neurons and neural development, I'm going to shift gears to the last story of the roundup, which is actually using a model system that we don't talk about too much on the show, but of course has a tremendous regenerative capacity, and that's the lizard tail, the tail of the lizard. So, as we know, lizards have an amazing regenerative capacity. Some of the other model systems that we've talked about on the show, like zebrafish, also have these, really amazing regenerative approaches and things that we're always trying to harness in the field, and ultimately trying to figure out how we can grow back our own limbs, kind of like what lizards and zebrafish can do, right? But the problem, and I actually didn't know this, that when lizards regenerate their tails, they do it imperfectly. So they're not able to regenerate their tail to a form that they're born with. And in fact, the tail that's formed through the regenerative process is it's really filled with cartilage, and that's not how it normally is born with that particular tail, right? And this kind of has to do with sonic hedgehog signaling. It's really important in tail regeneration in zebrafish. Um, so these folks at USC, University of Southern California, right down the road from me here in Los Angeles, um, in the lab of Megan Hudnall, they were actually introducing embryonic neural stem cells from the lizard back into the tail after the lizard is in the process of regeneration. And what they saw here is after introducing these NSCs that were modified to actually be knocked out for hedgehog signaling, which I told you in the beginning is important for tail regeneration in the first place. Um, for some reason, they're actually able to grow back in a more normal, natural way, like in a way that the lizard is actually born with, you know? So it's a story that's, I need to take a deeper dive into this, but apparently these smooth and knockout embryonic neural stem cells can, alter the formation of cartilage, which I said was found in the regenerated tail, but not in the natural born tail in these lizards. So it's an important kind of milestone in helping us understand why regeneration takes place the way it does in this model system. And if you think about it kind of super ultra long term, right? If we're thinking about regenerating human limbs and human organs, we want to be able to regenerate them to the way they were there naturally, right? The way the form of the organ or limb that you were born with, ideally, just restore it back to how it was. It's the Wolverine scenario, right? Perfect regenerative capacity. So we're not even close to that for humans, but perhaps at least in lizards, we can make their lives a little bit better and restore their tails back to the way they used to be normally, as opposed to these kind of imperfect amalgamations of the lizard tail. And it's a cool dev bio story. I'm always a fan of these cool model systems, as you know.
Yeah, you love these systems. You love the Evo Devo. You don't see it in the funding acknowledgement for this, but I have to say I'm speculating that this is funded by our future lizard overlords from outer space, because I mean, who the hell cares about regenerating lizards? That's what I'm saying. No, I'm kidding. It's very fundamental insight, and it's very important. And for me, just looking at it, cursory look, I will admit, but I'm really impressed by how, when you look into another system, just how foreign it is, and how they have these tools that you just didn't even know you could do. And you're like, wow, could I try that? Like here, they got the NSCs that they transplant. I see they got them from parthenogenetic lizard embryos, so that they could get a clonal match. Like, who knew? That's like a thing? I guess that's like de rigueur in the lizard systems, however many people are operating that. But like, I would just wonder, when you go, when I get a chance to do a sabbatical, that's what I want to do. I want to go to a model system that's totally whacked out of my wheelhouse, so that I can maybe try and incorporate some of these different ways of thinking. Because that's the key here, not just about the tech and the assay, but also just the mechanisms that go on in lizards, our future bosses.
I, for one, welcome our future lizard overlords. Okay. Well, you're right. I think I totally agree with you. We had Ken Poss on the show, who's an extraordinaire with all things zebrafish regeneration. We even talked about the Komodo dragon with Benoit Bruneau. Remember that? So there's really a need to better understand some of these cool, unique model systems. But I totally agree with you. When it comes to the actual genetics of doing the CRISPR in lizard embryos and lizard NSCs, I can't imagine that's easy. I don't know anything about the lizard genome. Maybe it's super well characterized, and maybe CRISPR is very easy and accessible in the lizard, but my gut feeling says it's not. So props to these folks for taking a stab at a really unique model system here that's very relevant for what we study in regenerative medicine.
Absolutely. Maybe we can talk to Kate. I mean, if anybody has a line with the lizard overlords out in space, it's her. But we'll see. We'll get to that. Before we do, I have a quick message from STEMCELL Technologies. Do you work with human pluripotent stem cell derived cardiomyocytes? Use STEMCELL's STEMDiff cardiomyocyte media and supplements to differentiate, enrich, expand, and cryopreserve functional HPSC-derived cardiomyocytes. STEMDiff cardiomyocyte media is compatible with human embryonic stem and induced pluripotent stem cells, and the resulting cardiomyocytes can be used for disease modeling, drug discovery, and cardiotoxicity screening. Learn more at www.stemcell.com/STEMDiff-cardio. All right, everybody. On this episode, we have a very special out of this world guest, Dr. Kate Rubins, who was fellow principal investigator at the Whitehead before she left that gig to join NASA, and is now an astronaut, been to space a couple times. Her research focus was in cancer biology and genomics. She's now a NASA astronaut and microbiologist, has completed 2 expeditions to the ISS, that's the International Space Station, spent 300 days in space, became the first person to sequence DNA in space. That was about 5 years ago, and grew cardiomyocytes in collaboration with Arun, my co-host, in Joseph Wu's lab at Stanford. Performed PCR and microbiome experiments while in orbit. She did it all. She was at the bench in space. I mean, can you believe it? She has a PhD in cancer biology from Stanford University and studied viral diseases as a fellow principal investigator at the Whitehead Institute. Dr. Rubins, thank you so much for joining us on the show today.
Absolutely. It's wonderful to be here. And I'm just going to enjoy catching up with Arun. When we normally work together, I'm not on the planet. So this is actually going to be a lot of fun.
Absolutely. Thanks so much for being here, Kate, and taking the time out of your crazy busy schedule, I'm sure, traveling back and forth in space to actually join us here on the show. I mean, I got to say, you probably have the best out of office message of all time, which when I received it back in 2016 was, quote unquote, I'm off the planet and unable to respond to your email. So you can't get much better than that, right?
Y'all are welcome to use that.
Okay, I might have to do that. But before you actually started taking business trips to the ISS, and you were, and of course you still are, a trained virologist with an amazing academic background, as Dalen just alluded to. And so since we are a cell biology show, tell us a little bit about your biomedical research road before becoming an astronaut. In particular, what inspired you to go into virology specifically?
Yeah, I actually started in public health in high school, and there was a program through the county public health department that had high school students get educated in HIV, and we learned from doctors as well as public health experts. We had meetings with patients, and what I thought was the coolest thing is we got to meet actual researchers that were working on HIV. So once we really learned about the biology and could be good educators, we went out and did peer education for HIV prevention. And I just thought that was a really neat experience. I was fascinated by the work that the scientists were doing. This was in the mid-90s when we had protease inhibitors for HIV, but we didn't have much else. And so the viruses were evolving resistance. And that idea that there's this dance between the virus and the immune system, and the virus does one thing, and then the immune system counteracts that, and then the virus can actually evolve resistance was very interesting. So when I went to school at UC San Diego, I started working in an HIV lab at the Salk Institute looking at integrase inhibitors. And this was kind of an exciting time to be doing that. So we were looking at both chemical compounds that would be inhibitory for the virus, as well as understanding mechanisms of HIV integration into the genome. And I think that's where I got very interested in genomics and what's going on with the host cell and the virus and the interaction between those two. I've studied that pretty much my whole life. I've just changed the virus family a few times.
Yeah, Kate, reading up on your history, it's hard not to get pulled into a rabbit hole containing the achievements of one of your mentors, Patrick O. Brown, who, to me, in my opinion, he's like the archetypal rebel thinker. Just in brief, he made amazing contributions to science, from retroviral replication to taking part in like inventing and formulating microarrays. Then on a sabbatical, this is the best for me, on a sabbatical just over a decade ago, he cobbled together the fabric of what's now known as Impossible Foods. I mean, I got some Impossible Burgers in my fridge right now. So the reason why I mention him is because with a mentor like that, who just is all over the place and beyond, It's not hard to believe that a doctoral student would see outer space as like a career path. I think most people would never even consider it. But do you think that there's a part for that? You know, the unbridled imagination of a mentor can unleash that spectacular ambition? Or is it, I mean, in your case, at least, you can only speak for yourself, or in your case, was space always in your DNA?
Yeah, absolutely. Pat was an incredible mentor. So I was really lucky. I had three mentors during my PhD thesis. I had Pat, I had David Relman, also at Stanford, and Peter Jarling was at the U.S. Army Medical Research Institute for Infectious Disease. So Pete really brought the Ebola piece of it. David Relman is a microbiologist. And he and Pat together have published a number of really exciting things. And Pat was just such a great mentor because he would decide what he was going to do. He'd have some crazy idea. When I first started in the lab, he's like, why are we paying to access journal subscriptions? This needs to be free to the world. You guys are only going to publish in open access journals. And all the grad students in the lab kind of smack our foreheads and go, oh my God, but we want this Science or Nature paper. And he's like, no, you're going to publish quality research and it's going to be in an open access journal. And we're going to build this open access. And this was before Public Library of Science was started. And he started that. And so I think to watch somebody say, this is what I want to accomplish, and everybody around him going, that's nuts. And then he just goes and does it anyway. And it was sort of the same thing with Impossible Foods. So I think that did give me the framework to say, hey, if you decide you want to do something, even though it really does sound like the most, when I was in high school and I said, I want to be an astronaut, people are like, yeah, you can't do that. This isn't a job that people go get. Like this, good luck. You're not a fighter pilot. I think I did have that framework where somebody's like, this is what I want to do and I'm going to make it happen.
Yeah, so let's skip forward a few years, and here you are as a NASA astronaut after all that training in virology and microbiology, right? And so I think what a lot of people don't realize is that the ISS, the International Space Station, is actually a research laboratory, right? A lot of our listeners may not realize that one of the primary functions of the ISS is for research purposes. And in fact, a lot of what you did in your daily work as an astronaut aboard the ISS was to conduct experiments for a whole range of research projects. And some of them, actually, like our iPS cardiomyocyte projects from Joe Wu's lab at Stanford, are biomedical experiments, and other are physical or material sciences experiments, right? So, I mean, tell us a little bit about a typical work day aboard the ISS, the International Space Station, and how you actually incorporated all of these research projects into your daily schedule, and maybe a little bit also about the capabilities and the equipment, like the microscopes, cell culture hoods, et cetera, that are actually there on space station for research use, because a lot of people don't realize that this is a pretty advanced laboratory.
Yeah, you see videos of astronauts, floating around and doing spacewalks, and we talk a lot about launches and landings. If anybody's ever watched a SpaceX launch, it's super cool. But really, what we do spend the most of our time on is laboratory work. And that actually is just as cool as the other stuff because you're doing it while you're floating. So your lab on the ISS, just to give you a sense, is the ISS volume is about the size of a 747. So we have multiple modules, and a lot of these modules have equipment that's devoted just to scientific research. So it's not just biology. Of course, we are doing material science. There were some really cool chemistry experiments, fluidics experiments. You don't have buoyancy-driven convection in microgravity. So you can look at materials properties in a completely different way. The physics experiments are awesome. Cold Atom Lab, there's a great paper describing Cold Atom Lab from last year that folks like physics. It's really cool. Of course, the biology was the one that I'm the most interested in, and the facilities there are quite good. It's really started to be equipped much more like a standard US lab. We do things a little bit differently. Of course, everything floats. So there's sometimes some very small problems that turn into big problems, like how do you pipette in space, or what do you do with your pipette tips? You can't just use gravity to propel them downwards into a little trash bag busted back on your lab bench. So there are a lot of things that we have to solve in terms of how do you do a laboratory setup, but the capabilities are really incredible. So it's spread out over several modules. We have at last count, I think I counted like 3 or 4 different microscopes on board. Some of them are fluorescent microscopes. There's confocal microscopes. We've got incredible tissue culture capabilities. And really, those were pioneered by your experimenter. And that was the first time that we were doing long duration cell culture on board the ISS. So we have a glove box that's dedicated to cell culture. It's not a laminar flow hood. It's actually a full glove box that you put your hands into. And we've got, incubators, centrifuges, everything that you would expect in a normal molecular biology lab. You just are going to do things a little bit different in terms of your fluid handling because it's all in microgravity. Everything's in free fall. So your experiments are floating, you're floating. That's what gives us these cool properties that we can examine in microgravity. The entire lab is in that microgravity environment. Just creates a few logistical challenges.
Yeah, you talk about, how you've had to adjust the framework there to suit, space and microgravity. And it gets me thinking that maybe that this is the future of science in some ways. We had Alison Watry on the show a while back, and it was right after he had recovered an ISS-bound experiment that had come back to Earth. And it got us talking about how experimental setups are due for an overhaul. And you talked about how Arun's experiment was pioneering because you had to figure out a way to make it work, right? And what we were talking about with Alison was that the automation would be essential for future labs. And it was more of a framework we're talking about on Earth with, a bunch of boxes and you're all doing it remotely, robotics. But, as someone who's classically trained, who's come up amidst the whole whirlwind of biotech and robotics and genomics and all the technology that's now at hand, but also you're really on the bleeding edge and have to visualize and imagine how we're going to practice science in the future and in these alternate environments. So how do you envision the actual, doing of science changing as we move forward? Do you think that it's going to look radically different? Or is it like you described? It's going to be tweaks of the things that look familiar here on Earth.
Yeah, I think from a micro, kind of a microgravity perspective, I imagine we're going to continue to build our capabilities. We've had labs on Earth for 100 years that have been doing microbiology. We've really only started to do kind of this more higher throughput molecular biology on the space station in the last 20 years. Have we even had that capability? I think we're going to continue to improve on that. And it's such a unique aspect to be able to tweak gravity in your system. We can't do that in labs on Earth. So I think there's a lot of things that we'll see in low Earth orbit that are still going to be capabilities. We may start to see those in things like commercial space stations. There may be applications for biotech or pharmaceutical companies that want to look at materials properties or cell growth or cell organization outside that constant 1G field. In terms of science in general, I saw this a little bit in my lab, even in the late 2008, 2009 timeframe. We were starting to transition. We still had a lot of wet lab scientists, but as we were thinking about hiring people, we were starting to transition more and more to people who could do data science, bioinformatics, large scale data set processing. And I think we've come to a point where we can generate so much data from a single experiment that the proportion of wet lab scientists are still always going to be there, but they now need partners or they themselves need to be able to process and handle all of this data. And so I think that we're going to start to see a lot of our scientific endeavor is going to just be more and more computational as we can generate increasingly huge data sets pretty cheaply from just a single experiment or two. So good bench lab scientists are always going to be in demand, but understanding how to design an experiment so that you can, in the classical experiments, you do something and maybe you get a result or you don't get a result on a Western blot. With a data-driven experiment, you're always going to get data. The question is it good data that's coming out the back end? So as a wet lab person, understanding that you will get information and making sure that you've designed the parameters of your experiment to give you that good information and then correctly analyzing it, I think is just going to be increasingly important in the future in ways that bench scientists in the 70s and the 80s never could have dreamed of.
Yeah, for sure. And I guess kind of piggybacking on that, in terms of the specific experiments that you might be the most excited about, I've ever since our project together in 2016, I've had the opportunity to connect with so many different folks in the space biosciences field, right? And it seems like everybody has their particular niche that they're trying to flesh out, figuring out how different cell types work in a low gravity context, right? And for sure, the ISS has become so accessible for basic biomedical research over the years, even in the last 5 years, but there's still a lot to learn about how the human body actually functions in space and the cells of the human body, right? So as somebody who's actually conducted a bunch of different experiments in space looking at different cell types, what sort of like current and future cell biology projects are you the most excited about? In part because they're cool to do, and also because they'll answer important questions about human biology in space.
Yeah, I'm really excited about experiments that are looking at multiple cell types and their interactions. I think you can look at, certainly the lack of gravity has various stimulatory effects, and you see things in all different cell systems that are different versus the ground controls. But I think where we're going to really see a large delta due to microgravity is when we start to have cell types interacting with each other. The way things grow and the way the fluidics forces shape our cell interactions are very different with and without gravity. And so I think when cells have certainly you can even see this at the macro level. When you look at humans, all of a sudden we adapt and we start working on the ceiling, and we change the way we locomote. We actually can see changes in the brain matter after somebody's been in space for a while. And so when you get down to the cell level, the way that cells are going to interact, for example, on a structure, on a scaffold, there'll be fundamental properties that will be different. I think membrane biology is going to be very interesting. And I think looking at multiple cell types interacting with each other and cell signaling will probably be a really rich field in the future. That's something that you can tweak with gravity. We can also look at partial gravity environments, and that's not something we've done a lot of, but that will be very cool. And it's very informative, particularly for our future exploration efforts to lunar and Mars gravity, where we're talking 1/6 and 1/3 G.
Wow, space on Mars. I mean, science on Mars. I'm waiting for that. I have to ask, because it's such a unique career path to be, this PhD in Stanford and the Whitehead and then NASA, right? So what's the next step? You can't spend the rest of your life as a space scientist. I mean, maybe you can, but how? Because it's a big shift, right? You go from studying viruses and genomics, and now you're the specialist in space doing all things cell biological. Do you think you'll find your way back to virology, or now you're studying microgravity, or you just, is it always a moving target? Do you have a path that you've tracked ahead of you, or you're just doing what's right in front of you?
I'm always doing things that I enjoy. And so I'm currently still on the active astronaut list. I'm part of the Artemis team that's working on developing our lunar exploration architecture. So the other thing that I get to do in my spare time is working on designing the next generation of spacesuits. And that's a little bit more to deal with the human physiology. How do you keep a human alive? How do you scrub CO2, generate oxygen, take care of thermal constraints? And then a lot about physiology. How are we going to move, protect people's joints? The way we locomote on the moon is this real bounding type mechanism versus floating in microgravity and moving around doing spacewalks with our arms, which is what we currently do on the space station. And so that's been a lot of fun, is to really look at things like human performance, maximizing capabilities and life support systems in the suit. I still do some virology. Actually, when COVID started, NASA couldn't train us in person anymore, and I found myself with a little bit of free time, and so found a group that was working on COVID diagnostics and got a chance to do viral diagnostics again for a little bit, just as a volunteer project, and I really enjoyed that. So I think probably, I will always keep an interest in virology and virus host interactions, and maybe we'll start doing some more research on that in the future. But certainly the microgravity research area is just exploding right now, and that's keeping us pretty busy with everything that we're doing at NASA with life sciences and cell biology.
Yeah, no harm in keeping your options open, Kate. And so I wanted to circle back actually to something that you brought up earlier, which is the commercialization of space. And that's, as space travel has actually become more accessible over the years, a lot of interest has developed in maybe leveraging space and microgravity for commercial opportunities, right? And this is in part, this is because of the unique properties of microgravity that impacts processes like important in materials manufacturing, sedimentation, for example. And even on the biomedical side of things, there's been some examples where microgravity has facilitated the development of biomedical products like protein crystallization during the Keytruda drug development process, for example. And bringing it back to stem cells, folks are actually starting to look at how microgravity might impact organoid formation or stem cell differentiation, all these things that could have translational value for cell therapy here on Earth. So what do you think about leveraging space for improved like biomanufacturing of products or maybe processes that couldn't be developed here on Earth? And what questions do you think we need to actually address and answer before biomanufacturing in space can actually become commonplace?
Yeah, I think a lot of it's in the early stages. A lot of these biomanufacturing questions are centering around things that are chemical or material processes. So the lack of buoyancy driven convection is really interesting, particularly when you're talking about fluid mixing. When you've got more viscous fluids, this can be really very interesting for certain types of metals and looking at melting temperatures. Combustion in space is very different. And so when you're talking about making a physical product, when you make that product in microgravity, there's a company that's looking at possibly manufacturing corneas. And if they can manufacture those in space, they could possibly have more spherical shape and less defects. And so these are still in the very experimental stage. Another thing that they're looking at is a particular way of manufacturing these Z-bland fibers, so these optical fibers, and doing that process in microgravity. Given the material composition and the way things, we spend a lot of time extruding these fibers from various manufacturing machines. And so these are things that can happen either on the International Space Station as part of commercial efforts or possibly in the future on commercial space stations. I think it's going to be one of these things where the field, people try a whole bunch of different things. Some things are not going to work better in space. And then some things may give you such a value that it is worth it to think about how you would scale up and actually manufacture that item in space. Some things may just be more in the basic understanding of the manufacturing process. So you may not manufacture that item in space, but you may refine your methods based on what you find in microgravity. I think from the biomanufacturing side of things, we're still early in the days of working on developing bioprinters. So we're sending a couple of those to space station, and we're going to start printing biological materials, different cell types, different scaffolds, that kind of thing. I think obviously the long-term goal is could we develop, organoids or organs and actually print those in space? Lots of things have to be answered in terms of like facilities. Could you maintain that level of cleanliness? Could you actually do a production level printing where you would then implant something into a human being? There's a whole lot of processes that have to be established, and that's of course going to be harder in space. But I think it's very cool that we're taking these first steps and sending these materials in space and actually working on doing biomanufacturing in space, and then analyzing what comes out and see if there is truly a benefit there. And then what can we learn about the basic biology? I think the discussion about the commercial applications is an exciting new field. That does not mean that we're abandoning the basic research. There's still so much to learn, and low Earth orbit right now, the only place that we really can effectively get rid of gravity or tune it as a variable. So we're going to keep that the basic research component around for a long time. And there's a lot of really cool fundamental biology questions that we can answer that don't have an immediate commercial application.
So yes, notwithstanding all those commercial applications, also, there's a lot of philosophical, technological, medical, what have you, justifications for having a presence in space and beyond. But there's the recent advent of space tourism has stirred debate as to whether or not commercialization of like the tourist element, at least, is the best allocation of resources. So as a scientist, what's the science response to that? Is there a science response where you say there is an upside for humanity writ large, particularly, I mean, upside for those people on the opposite end of the spectrum is like the Bezoses and the Bransons. What do you say to those people to say, yeah, all these civilians going up to space, there is a long game upside there, or isn't there?
Yeah, I think, some of the big questions about this is resources, and is this how we should be spending our resources? And, I do think that people worry about spending money in space. The thing is, though, that the money's not being spent in space. There is actually no currency in low Earth orbit. So, I think it is a good question of where should we be putting our time and our resources. But really what a lot of that money is going to is to develop the space industry. And so a lot of these players in the commercial market are either startup companies, or companies that are working on both NASA contracts and space tourism. I personally, the billionaires can do what they want. That's their money. I don't know that we're going to get huge benefit to humanity by sending a bunch of billionaires to space. I'm much more excited in making space accessible to everybody. And so unfortunately, I think the way that you have to do that sometimes is it can be very expensive to start to do something. When you look at airlines at the beginning, it was so much more money than any of us could have afforded to take a transatlantic flight. And now this is something that we can do. So I think it's good to have people pave the way if they want to offer up their resources and it's not government resources and taxpayer dollars to do it. That's fine with me. But I would like to see this be open to everybody. I don't want this to be an exclusive club that's for billionaires only. The benefit that I really see in terms of for just everyday people, and I am one of these everyday people. I'm not a billionaire. I am just a normal, regular bench scientist who happened to be an astronaut. But I get the chance to go talk to kids. I get to reach out to people who are maybe interested in being scientists or astronauts and talk to them about space exploration. And sometimes people will want an astronaut to come talk at their school, or they'll want to have a discussion with an astronaut because they said, well, you've been to space, tell us about that. And we end up talking about science the whole time and the cool things that you can do in microgravity and the way that science is exploration. And so I think that there is a lot of value in inspiring kids to go into STEM fields. And I think that's where, if they look at this and go, hey, maybe right now you have to be an astronaut with all this training or a billionaire, but by the time I'm old enough to get my space driver's license, I could actually do this. I think that's a pretty cool thing. And it's something, if we really want to venture beyond this planet and we want to start exploring the rest of our solar system, we are going to have LEO, low Earth orbit, move more towards commercial applications and to the space tourism. And this is really going to allow us as the exploration side of things, the government side of things, where we can take those risks to move further and to go to the moon and to go to Mars.
Sending Shatner up there. It's amazing. Wow. Well, this has been great, but we're not going to let you go yet. Just got a couple of peripheral questions here. So first, you are a scientist and an astronaut. If you weren't either of those things, what would you be?
So little known fact, I was actually an English literature minor in undergrad. I really enjoy reading. I don't know that I would have gone into English literature as like a full-time profession. I think I would have been an academic in some way. I took a very cool class that was kind of computer machine human interface in undergrad. And I thought that was very cool. I probably would have done something with how we interact with machines and the sort of the intersection of cognitive science and computer science and behavioral and psychology issues with humans. I think that's very cool how we interact with our not our man-made environment, and that's becoming increasingly important with looking at how we interact with our devices. And it's actually one of the things we do a lot when we talk about our interaction with the spacecraft is what are human factors, what are ways that we can design spacecraft and spacesuits so that we reduce error and we increase our mission success capabilities.
Wow, I mean, the way you've thought that through, it's like you almost did that. I feel like you kind of like half did that, but then you also became an astronaut. All right. Next, what is one piece of advice that you'd ever been given that you'd repeat to someone behind you, either professional or not?
I think professional advice, and particularly for students, grad students, undergrads that are starting to work in the lab research it's awesome, it's exciting, and it can also drive you nuts, like when you're at the bench and your experiment's not working for the 50th time. And so I think it is, pick something that people always say, pick something you're passionate about. Like no kidding, pick something that you wake up and you're like, I want to go answer this freaking question. And I'm going to put up with my experiment not working 50 times because I'm so stubborn that I want to get the answer to this question. And if you've got a question that you want to answer, then you're pretty happy, even when things aren't working, to spend your life, trying to figure out, the answer to this question, solve this problem. So pick something that grips you. And a lot of times when you really talk to people, they'll have something that, maybe around the age of 10, 11, or 12, that seems to be the time where people are like, I found this thing and I got really interested in it and I haven't been able to let it go. So if there's a, question that they were thinking about a lot when they were a kid, or there's something that they're seeing in their current research, and it's like, why is nobody else answer this? A lot of times I think grad students maybe think they're a little bit too unaware of their own value. Think, well, nobody's answered this because it's too hard, or maybe somebody's answered it and I just haven't figured that out yet. Sometimes people haven't answered it because they haven't thought of it. They haven't had the time or the resources. And so if it's a cool question, I think try to answer it. And sometimes the way to do that too is to combine a few different kinds of fields. So I'm giving you two pieces of advice for free. The intersection of two different fields can be incredibly exciting, and we get very siloed and very in depth into our particular chosen field. But if you say, hey, I'm going to put this thing together with natural language processing, let's look at different kinds of algorithms to understand this data set, or why don't I combine this chemistry piece with this very interesting cell biology piece, that kind of thing. If you're willing to put in the time and the effort to work in two different fields, can be incredibly rewarding. And that's where you make your career, and that's where you have fun spending your time.
That's where you blaze your trail, guys.
Mm-hmm.
Okay. Finally, I can't let you go without this. This is kind of a personal question, but I'm sure everybody's wondering the same thing. What's the best thing about it? Is it getting up there? Is it the ride down? Is it being on the float? I mean, if you had to pick one thing, or tell us all of them, what's so great about going to space?
I think probably the best thing is just the opportunity to see Earth from that vantage point. I didn't think it was going to be that cool before I launched. I was like, yeah, I've watched all the movies and I've seen the Earth from mission control. And there's something about seeing it with your own eyes. The Earth is so much brighter than we can really see in TV or movies or film. It's maybe the way the human eye processes the dynamic range of light, but it's like this glowing ball. And it's so much brighter than the blackness of space. And then you really can see a tremendous amount. You're looking at the continent level, but you can get these cameras and look down at the Earth. And so you get to see all these places on Earth that I'll probably never visit. Pretty much my favorite thing was just sitting in the cupola, which is our module that looks down at the Earth. And we've got these 7 giant windows, and you can actually tuck your whole body into the cupola in a little ball and just float around the Earth, do a whole orbit for 90 minutes. So I would put in my headphones, I'd get a good movie soundtrack going, and I would go around and do an orbit from day to night, and just fly over the planet and look at this gorgeous Earth, and kind of think about wow, how do we end up here? How is it so incredible that our planet supports life? This is an amazing experience to be able to see it.
Wow, what a trip. Thanks for joining us, Kate. This is one of a kind for us, so we really appreciate it. And I'm sure our listeners too are inspired by your words and just the images. I mean, I'm picturing this bright blue ball. Hopefully one day I'll see it. Me and Shatner both. He'll take me up the next time.
I hope so too.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on Twitter at StemCellPodcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. Really, guys, suggest some guests. There's really no guests who might be inappropriate. We got an astronaut on today, and I think that shows you the breadth of our interest and ambitions in recruiting guests. Thank you for listening, guys. You'll hear from us again in a couple weeks.